Today we will learn about protein synthesis. Wait a minute, I have a great idea. What is it? Let's go learn it in 2D. That's so smart, wow. Let's go.
Transcription is the first step in gene expression, in which information from a gene is used to construct a protein. The goal of transcription is to make an mRNA copy of a gene's DNA sequence. RNA is processed and forms the mRNA. Let's look at the bigger picture. Inside this eukaryotic cell, you can see the DNA, which is a polymer composed of two polynucleotide chains that coil around each other to form a double helix that carries genetic information. DNA in the transcription process takes place in the nucleus of the cell. RNA polymerase is an enzyme that synthesizes RNA molecules from a template of DNA. The RNA polymerase first binds to a DNA promoter sequence and then moves through the DNA template, splitting the two strands apart, reading one base at a time, and matching it with its matching base, which builds an RNA molecule made out of complementary nucleotides and an mRNA strand that is complementary to the transcribed DNA strand. And if we zoom in even more to the nucleotides for RNA, you can see that it consists of a nitrogenous base, a phosphate group, and a ribose sugar. Let's zoom out again. So after transcription, the transcript is released from the RNA polymerase, and then the mRNA strand is released. After the two unzipped DNA strands bind back together, forming the double helix, the mRNA is then processed to remove non-coding regions, introns, and leave the protein-coding regions, extrons. The exons then become close, cutting out the introns, which are released. The main strand later leaves the nucleus and enters the cytoplasm to go into translation. Translation occurs after transcription and is the stage where the mRNA is decoded to build a protein that consists of a specific series of amino acids. Translation takes place on ribosomes in the cell cytoplasm, where mRNA is read and translated into the string of amino acid chains that make up the synthesized protein. The mRNA binds to the ribosome, and then a transfer RNA, an adapter molecule composed of RNA, known as tRNA, carries the amino acid and binds to the start codon of mRNA. It later acts as the binding site for three bases, also known as the anticodon, which is complementary to the mRNA codon. The anticodon reads what the tRNA transferred and then finds the matching amino acid to pair, and this repeats until it reaches termination, which happens when the stop codon in the mRNA enters the A site. Now let's rewind a bit and show a scenario where mutations occur. Mutations are variations of patterns which involve changes in the sequence of nucleotides in DNA or changes in the number or structure of chromosomes caused by errors in genetic processing. There are many types of mutations, but the most common ones are point mutations, frameshift mutations, and chromosomal mutations. Let's start with insertions and deletions. These mutations can take place by inserting a nucleotide to a segment of DNA. This results in a base and amino acid change and possibly severe effects on gene expression or defective proteins. As you see in this side-to-side picture, the whole pattern changes, causing a change in the sequence. The same applies to deleting a nucleotide to a segment of DNA, which causes a change in the sequence and pattern of the gene expressions. A point mutation takes place on a single nucleotide in the DNA sequence. It is also called a base pair mutation or a base pair substitution. A point mutation that does not have any harmful effects and does not change the sequence of amino acids in the protein is called a silent mutation. A same-sense mutation is a form of silent mutation where a change in the base sequence of a gene does not result in a change in the amino acid sequence of the protein which the gene carries the genetic instructions. For example, if the codon GGG is altered to become GGA, the same amino acid, glycine, will be incorporated into the peptide chain, and this is considered a same-sense, aka silent mutation. A missense mutation is a mutated codon code from a single base pair substitution that leads to a different amino acid. A nonsense mutation is a mutated codon that is a premature stop codon rather than specifying an amino acid like other mutations. Frameshift mutations are slightly different because frameshift mutations are where one or more bases are inserted or removed from the DNA sequence. This can have a more severe effect because it typically shifts the reading frame of genetic messages, changes every amino acid following that point of mutation, and can alter proteins to paint where it can't function. Now that we learned about DNA mutations, let's go back.
Wow, our voices are back to normal.
I know, right? How crazy.
Hooray, I can't wait to see Mr. Paige.
Thanks for watching.